Esta conferencia comenzará a grabarse.
So as I was saying, my name is Javier, and I will be presenting today's webinar about Cytognos NGF flow solution for immune monitoring COVID-19 and other pathologies using the PIDOT and the immunoglobulin subclasses kit. I will start introducing ourselves, Cytognos, for those of you who might not know us. We are a clinical flow cytometry manufacturing company that works in the fields of oncohematology and clinical immunology, and we are focused in developing solutions for labs that work in those areas. We were born in 1996 out of the hematology department of the University of Salamanca in Spain. In 2006, we started collaborating with the Euroflow group in bringing its flow cytometry method to the market, including the antibody panels and the analysis software, Infinity. More recently, we incorporated a CE/BD instrument to complete our flow solution. And also, since the last year, we have started developing solutions for the immune monitoring market. And this part we will go through this webinar. But first, let's talk about the value of the clinical lab. The clinical lab plays an essential role in the healthcare system, as it provides the information that enable doctors to make the right diagnostic and therapeutic decisions for their patients. So, as clinical laboratory professionals, we have a direct impact on many aspects of the patient care, including making a direct improvement in the life of the patients. And of course, one of the fields where clinical labs can have a key role is in the monitoring of the patients. In order to provide this high-quality information, we need to address several aspects that have to do with mainly two things. In our labs, we always look to improve our laboratory efficiency, and for this, we will try to optimize resources, ensure the reproducibility of our instrumentation, minimize the subjectivity of our data analysis and reporting, and have a good training program in place for our lab staff. And also, we always have in mind that our work will provide better patient care, as by providing a timely and accurately laboratory testing, we get a quicker diagnosis that leads to faster and better treatment, which eventually translates into shorter hospital stays for our patients. With this in mind, from Cytognos, we work to help clinical flow labs in achieving these aspects, both providing better patient care and being more efficient in our workflow. And that has been our goal while developing flow cytometry solutions. After this brief presentation, in today's webinar, we will review the role of immune monitoring in pathologies such as cancer, autoimmune diseases, and infectious diseases. And here, we will focus on COVID-19 and will analyze how flow cytometry can help us better understanding this pathology. We will move on to our next generation flow solutions for these applications, where we will present you the PIDOT and the immunoglobulin subclasses kit, oriented as part of this complete solution. And I will show you a few files, including a patient file as an example. We might also have some minutes at the end for a couple of questions. Okay, now let's move to immune monitoring patients. We know that one of the traditional divisions of the immune system could be as lymphocytes, where we can find B, T, and NK cells, and myeloid cells, where granulocytes, monocytes, and dendritic cells can be included. Within these groups, we could identify more functional groups and have a deeper knowledge of the distribution of those subpopulations. But we also know that these are not closed groups. All the different immune cells interact with each other, exchanging information about the environment and regulating the function of the rest of the cells. In this sense, If any of these cellular compartment is affected, for example, in the case of an infection, the other cells of the immune system may be affected as well, and that's going to change the homeostasis of the immune system. That's why it's important not only focus on the target population of the disease that we are studying, but to analyze the environment so we can know the status of the immune system and obtain information from it. And in this sense, immune monitoring our patients before, during, and after therapy will provide us with a general view of the immune system and will give us details on how the different populations can be affected. And this can be especially relevant in cases of immune therapies, as most of them affect directly to the immune system. Immune monitoring also allows us to track the recovery of the immune system over time. And this could be very important in some diseases, as there are studies correlating a normal recovery of the immune system with a better prognosis for the patients. For example, after autologous bone marrow transplantation. We can also follow the pathological population after the treatment has finished and compare it to pre-treatment levels. So, monitoring our patient will bring us several benefits. It will improve our knowledge about the state of the patient. It can bring prognosis value in the dynamics of recovery and will allow us to make clinical and therapeutic decisions according to this knowledge. All of this in the end resulting in a better and more personalized care for our patients. And as most of us here are cytometrists, flow cytometry is a very good technique for this purpose, making the monitorization very specific and reliable. There are more and more studies highlighting the importance of characterizing not only the target population, but also the homeostasis of the immune system. Here are just a few samples related to the fields of hematology and autoimmunity, as monitoring the recovery of patients after treatment and knowing the tumor microenvironment can provide us of vital information about the response and survival of our patients. And from this point, we're going to focus on the immune monitorization of COVID-19 patients. We're going to make a review of some of the works that have been published that highlight the benefits that bring the monitorization by flow cytometry of COVID patients. All the information that we can obtain by this technique, and also how patients can directly benefit from it. So here we want to refer to the review that was recently published by the group of Andrea Kosaritza, where you can find most of the articles that we are using in this webinar. The first studies focusing the circulating immune system by flow cytometry in patients with COVID-19 were carried out in China, and they showed a significant difference in cell distribution in patients compared to normal distribution ranges. As you can see, for example, here, the total T lymphocyte count was diminished compared to the normal range. On top of that, these changes were especially strong in severe cases of COVID-19, and the reduction of normal counts could distinguish patients with a severe form of the disease from more moderated cases. So, the first conclusion we can get is that immune monitorization of COVID-19 patients by flow cytometry could help us distinguish between moderate and severe cases of the disease. These findings were reproduced by other groups, and in this work, they correlated this reduction in the circulating population with clinical parameters of inflammation. We can see how levels of inflammatory markers like the erythrocyte sedimentation rate, the C-reactive protein, and the interleukin-6 were higher in patients with low counts of Tcells, and this correlation is stronger when referred to the decrease of CD8 Tcells. As you can see, the correlation is negative between the high number of Tcells, CD8 in this case, and the clinical parameters. So as you can see here, changes observed in the distribution in patients are directly correlated with some clinical markers. As we said before, monitoring over time can also bring us very relevant information about the progression of the patient. In this article, we can see that by tracking the immune system of COVID patients, we observe that those patients who recovered normal levels of circulating leukocytes, like you can see here, were able to completely eliminate the viral load, while patients that after 10 days still presented reduced counts could not eliminate the viral load. This brings up the prognosis value of making immune monitoring by flow cytometry. In this research work, it was found that in lung aspirates of COVID-19 patients, the distribution of the cells was severely altered, and way higher percentage of myeloid cells were present in patients compared to healthy control. And as in work before, these changes were stronger in severe cases. So we have to take myeloid cells into account to get deeper and better information about the development of this disease. This work connects with the idea of the previous one, as here the group categorized patients using two parameters: the level of circulating immunoglobulin G and a neutrophil leukocyte ratio. Interestingly, they found that patients with a more severe form of the disease concentrated in this group that presented high titers of immunoglobulin G in the serum and also high count of neutrophils, showing us again the importance of studying not only the lymphocytic compartment but also the myeloid cells. Also in this same work, they found that the dynamics of the serum immunoglobulins were different between patients. As you can see here, they found that there was a peak in immunoglobulin M in patients that occurs with a more severe form of the disease. Now it's important to notice that one of the treatments proposed and that is already being applied in some countries is the infusion of hyperimmune serum from people that have gone through COVID-19. This infusion will make us unable to monitor our patients at serum level. However, by flow cytometry, we can monitor at cellular level, so even when we are altering the serum immunoglobulins, we can still check at the cells that produce these molecules in the patient. And finally, we come back to where we started, to the review of Andrea Kotsarica. After reviewing these different articles, the group pointed how we are still missing a lot of information about the disease, and we wanted to comment some of the conclusions they reached by the end, like the role of the innate immune cells, that, as we have seen, is very little is known of their distribution, and how this knowledge could help us get ahead of the virus. Also, we need a deeper understanding of the role of the different subpopulation of CD4 and CD8 T cells. If the changes that we see in the patients are produced in specific compartments of these T cell groups. And also the kinetics of the humoral response, along with the importance of the memory B cells and plasma cells in the development and the control of this disease, as we also need to understand what happens not only during the infection, but also after it with the antibodies and the memory cell to prevent reinfections. With all this data in our minds, we can now discuss how our technique should be in order to successfully monitor COVID-19 patients by flow cytometry. As we have seen, we need sensitivity in order to be able to compare the data of our patients with a robust database of normal samples with normal reference ranges. It is very important as well that our technique is standardized and by so reproducible, as we intend to follow one patient over time, and we want our data to be comparable from one day or 1 week to another. Ideally, we will need to identify as many populations of lymphoid and myeloid cells in an efficient way, without the need of a huge screening panel full of different tubes, so we can easily introduce this application into our lab routine. And as we just commented, we should be able to detect expression or distribution changes at cellular level, so we don't have to worry about different treatments affecting our monitoring. And to cover all these aspects, I'm going now to introduce Cytognos Next Generation Flow solution for immune monitoring. This solution will cover all the points we have just seen in order to facilitate the implementation of this new application in your lab, in a way that you gain efficiency and also improve the care of your patients. This solution includes antibody reagents, which in this case are going to be the PIDOT and the immunoglobulin subclasses kits, standardized operation procedures developed and validated by clinical flow cytometry experts, the flow analysis software Infinisight, and Omnisite or CE-IVD instrument. And all of these provided with the training and support you need to get your assays up and running. One of the two kits that conforms this solution is the PIDOT kit. With this 8-color panel containing up to 12 markers, we will be able to get a complete identification and enumeration of the circulating lymphocyte and myeloid populations. We will see this in more detail in the demo, but with this panel, we will be able to identify most of the population of circulating leukocytes. In a first step, we can identify B, T, and NK cells and different myeloid cells. At a second step, we can classify the two main T cell subsets, such as cytotoxic T cells using CD8 markers, the CD8 marker, and helper T cell with the CD4. And here we will analyze the distribution of naive, memory, and effector cells in these two populations. We can also identify different B cell subsets, since these panels include the markers to study pre- and post-germinal B cells, post-germinal center B cells, and here both unswitched and switched B cells. And what is also very relevant is that within the myeloid compartment, we can detect the different types of granulocytes as neutrophils, eosinophils, and basophils, as well as monocytes, both classical and CD16 positive monocytes, and also we can identify the plasmacytoid dendritic cells, having so a detailed landscape of our myeloid cells. With this panel, we are identifying over 20 different immune populations in a very efficient monotube panel. And also, with all the markers present in the tube, we will be able to detect any change that is produced in the patient at cellular level, which will give us the information we need to make better clinical decisions. We also talked about the importance of having a robust comparison to detect changes in our patients in a sensitive way. And to address that, as we previously remarked, our solution is based in the work of the EuroFlow group. With our solution, using the PIDOT panel, we can compare the data obtained from our patients with the published normal reference ranges that were developed by EuroFlow. And these ranges are presented specifically segmented by age. This can be very important, as we all here know the distribution of the different populations of the immune system change throughout our life, and we can have different number of lymphocytes and myeloid cells compared with a child or with an older person. And so, in this way, we're going to compare every and each population that we identify with our panel with their normal reference range. And for some population, as for example the plasmacytoid dendritic cells, these ranges could be very difficult to find. All this will avoid the need of validating and developing our own control population, making it a very reliable and fast to implement technique. Also, you have all this information published with open access if you want to get deeper into this. Another advantage of our solution is that working with the Lymphocyte analysis software, the access to this information is very fast and intuitive, as using the automatic gating and identification tool of Lymphocyte, we have access to the automatic report. This report will present us the relative distribution and the absolute count for all our different populations, and we can compare it with the normal ranges of their specific age. There are 14 age ranges to cover from childs to adults, and this will allow us to make our comparison in a very precise way. This report also highlights the relevant information found in our patients, making the transmission of the data very efficient in our lab. Like for example here, you can see that the post-germinal center are lower than the normal ranges, and they are presented highlighted. Now, to see all this in action, let's jump to Lymphocyte. Okay. We're going to start with a normal sample, so I can show you all the populations that we can identify with the PIDOT panel. This sample is already analyzed using the automatic gating and identification tool included in the Lymphocyte software. This tool compares our sample with the EuroFlow databases and automatically assigns all the events that matches with normal populations present in the database, making our analysis faster as we just have to focus on analyzing the events that are different from normal cells. You can also see that the software presents us some multiparametric diagrams that allow us to check in a simple glance if we lack any of the populations in our samples. For example, here there are presented the different leukocyte subsets and where they should be placed, and we can see that in this particular sample they can all be identified. With the automatic gating and identification tool, our objective was to make the analysis more agile and to bring the important information to the front. Now we're going to go through all the populations present in the sample. And let's start by studying the lymphocytes. For studying the lymphocytes, we can start by using this diagram of CD3 against CD19 and TCR gamma delta, where we are going to be able to identify our Tcells and our Bcells. And here in the double negative area, and also using CD16 and CD56, we can identify our NK cells. Okay, now going a bit more in detail, we are going to analyze more deep our B-cell population. As I said, we are going to be able to identify both pre-germinal and post-germinal center B-cells. And for that, we have here our CD27 marker that is going to differentiate these two populations. Here in pink with CD27 negative, we have our pre-germinal center B-cells and our post-germinal center B-cells that are CD27 positive. And here in this population, using the immunoglobulin, the surface immunoglobulin D and immunoglobulin M, we can distinguish between the B cells that have gone through class switch recombination, that have lost the expression of both IgD and IgM, and the B cells that are still unswitched because they maintain expression for both markers. And we can even identify a small population that are post-germinal center because they are CD27 positive and they express only, and in a very bright way, IgD. So now, moving to our T cells, we can start identifying the two main subpopulations of T cells that we can find in circulation, that are our T helpers with the CD4 marker and the cytotoxic CD8 T cells. And we can also distinguish between the gamma delta T cells and the alpha beta T cells. If you remember, one of the points we found important in the review of the articles was that we need a better understanding of the subpopulation of T cells. And for that, we can use this plot of CD27 against CD45RA. Starting by the CD4 T cells, with this plot and these two markers, we're going to be able to identify different stages of maturation inside our T cell populations. Those that maintain the expression of CD27 and CD45RA are going to be our naive CD4 T cells. After losing the expression of CD45RA, we can identify our central or transitional memory T cell, CD4 T cells. And here we're going to have two different effector populations, and both have lost the expression of CD27 and that are distinguished by the expression of CD45RA. Are effector T cells that express the CD45RA and the effector memory T cells that are negative for both CD27 and CD45RA. We can find similar populations or subgroups in the CD8 T cells. We have here our naive T cells that, as we said, are bright for CD27 and CD45RA, or central transitional memory CD8 cells that lose the expression of CD45RA but maintain the expression of CD27, and our effector populations that in the case of CD8, we're going to have three of them. Our effector memory that are negative for CD27 and CD45 array, and the two populations are of effector CD8 that can be distinguished by the expression of CD27. Okay, so now that we have gone through all our lymphocyte population, let's jump into the myeloid cells. Starting by the granulocytes, we can identify using these panels the eosinophils, neutrophils, and basophils population. And for this, we can use mainly these two diagrams for the expression of CD45 against the site scatter, where they can be distinguished, and also for the clear identification of the neutrophils, we have the plot with the expression of CD16. In addition to the granulocytes, as we said, we're going to be able as well to identify our two populations of circulating monocytes, the conventional traditional monocytes and the CD16 positive monocytes. And they are very easily distinguishable using the diagram of CD45 against CD16. And as you can see, these populations are of similar height, but the expression of CD45 is a bit brighter in the case of the CD16 positive monocytes. Oh, sorry. And to finish, we can also identify our plasmacytoid dendritic cells, and they can be distinguished as they are going to be in the area of the lymphocytes or in the transition between the lymphocytes and the monocytes. They are going to be negative for the expression of CD3 and CD19, but they're going to have a mild expression of CD4, as well as they are going to be very bright for CD45 RA. As we have seen with this panel, we are not only studying in detail the lymphocytes, but we also obtain very valuable information from all these myeloid cells, getting the knowledge we need to better follow these populations in our patients. And now that we have finished the analysis, we can move to the automatic report that Infinisite generates based on the results of our analysis. Here, as you can see, the report presents us in a very intuitive way the absolute count and the relative frequency of our sample compared to the reference range of their specific age. And for this case, we can see that it is applying the range of 30 to 59 years. We can now use these normal ranges to make sure if our patient presents any alteration in the distribution of this immune population. As for example, we can check if our Bcells are within normal ranges, both in absolute counts and in frequency. And you see here presented the result from our patient compared to the reference and seems to be normal. This is a normal case and all seems to be fine. But now let's move to a COVID-19 patient. I'm going to close this. Okay. And now I'm going to open with this. Sorry. This is a sample from a COVID patient that was taken at the day of the hospitalization. So we will call this the day 0 sample. Please take into account that this is a single file from a specific patient, and that the difference that we find in this sample might not be present in others. Our goal here is to be able to find the differences in our patients. To not make this very long, I'm going to focus on showing you some changes in the expression of a couple of markers. But you can see here in the column of the alerts that the software is already noticing that we have differences in this sample compared to the normal database. But as I said, let's bring the Bcells and analyze them with the same plots that we used last time. Sorry. Here in the plot of CD27, we can observe that both post-germinal center populations, the unswitched and the Bcells that have gone through immunoglobulin class switching, are expressing very brightly CD27, the CD27 marker. This could mean that the Bcells from this patient are taking a more producing phenotype and are converting into plasmablasts to start solving the infection. Moreover, the unswitched Bcells seems to be focused on the expression of IgM, as we can see in this plot of IgD versus IgM. We are being able to monitor at cellular level the changes that we saw that are produced in these patients, and we can now understand how are the serum levels changing from another perspective. Another change that we can observe in this patient is the expression of CD16 in neutrophils. So I'm bringing it up. And here you can see that there is a group of neutrophils that seems to have reduced expression of CD16. CD16 negative or low expressing neutrophils are more immature cells that are being released from the bone marrow. The immune system seems to be fighting against the virus and it's bringing cells at a very fast pace. And now let's move to the report to check if there is any alteration of the normal distribution of our populations. Okay. Now we can see that some of the immune populations are highlighted, and this means that they are out of range, either in the absolute count or the relative frequency compared to the normal database. We can see, for example, our pre-germinal center Bcells are reduced compared to the normal reference range from its specific age segment. And we can also see here that our CD27 negative effector Tcells are augmented. We can also check that the distribution of different B and T cells is altered compared to the normal ranges. In addition to this that we find in the lymphocytic compartment, we can observe that the myeloid cells are also very severely affected. All the granulocytes, monocytes, and dendritic cells are reduced in this patient, as you can see here. And some of them are very and severely reduced compared to the normal reference range. So it seems that checking these populations might indeed be very important for these patients. Okay, let's move again to the presentation. As we have seen, with Cytognos solution, we can monitor very precisely the changes that occur in a patient. And these changes can be in the expression of the markers present in the panel, as we have seen in this patient, where the expression of CD27 is higher than in normal samples on Bcells, and the reduced expression of CD16 in neutrophils. But not only that, we can also check any changes that are produced in the distribution. Here we can see the report of the patient that we have just analyzed at day zero, the day of hospitalization, and we analyzed the same patient after 7 days in the hospital, when the patient was almost totally recovered. And as you can see, with Cytognos solution, it's very easy to follow any change in the distribution of all these populations that we study with the PIDOT panel. You can check how after 7 days, the numbers of CD8 T cells increased, and this was more evident in the memory and the effector populations. We also see that neutrophils were deeply reduced, and they still maintain the same after 7 days. On the other hand, monocytes recovered normal levels. And interestingly, in this patient, we can observe an important increase in plasmacytoid dendritic cells as well in basophils. As we are now able to detect these changes, we can follow our patient in a very efficient and sensitive way, and that gives us the chance to improve how we manage COVID patients. At this point, we have gone through how the PIDOT reagent kit is going to help us monitoring the patients. And now we are going to check the other kit that Cytognos offers for a deeper monitoring of the B-cell compartment, the immunoglobulin subclasses kit. With this kit, we are going to focus on the B-cells, and we are going to characterize different populations of immunoglobulin G and immunoglobulin A. Using this panel, we will be able to identify in both B and plasma cells the production of immunoglobulin G1, 2, 3, and 4, and also the production of immunoglobulin A1 and 2. In addition to this, we also studied the expression of IgM and IgD, and due to this, as we have discussed previously, the identification of the cells producing antibodies at cellular level will complement the measurement of soluble immunoglobulins at serum, and this will allow us to detect the changes that are produced in the cells even when the treatment alters the circulating levels of immunoglobulins, like for example, when we make infusion of hyperimmune serum. To detect changes in the patients in a sensitive way, we also have available in open access publications the 8 segmented reference ranges for both B and plasma cells that we can use to detect those alterations in the distributions that are produced in our patients and compare them with a control group of their specific age range. And now that we are approaching to a vaccine, the monitorization of the specific immunoglobulin subpopulations might give us the key information in the way of making a viable vaccine with a strong and durable response. And also, as we saw with the PIDOT, having these reference ranges available with specific age segments is very important in the case of monitoring the immune system due to the changes of the distribution that we present throughout our lives. And so we will have a very specific way to know how our patients evolving. Just to sum up, what today we wanted to show you is how we can address all the key points that we commented using the next generation flow solution proposed by Cytognos on monitoring not only COVID-19 patients, but also from other pathologies. We have seen that with our solution, we can compare our patients with an age-segmented database, where we have normal reference ranges that will make our detection of changes in the distribution very sensitive. We also discussed how, based on the Euroflow methodology, with their SOPs as one of the central points of our solution, we get the standardization and the validation needed to make our monitoring reproducible. This will allow us to compare samples with other centers and follow our patients over time. And finally, we also checked how we can identify a large number of lymphoid and myeloid cells and detect changes in their distribution or in the expression of their normal parameters at cellular level in a very efficient way. This will optimize our resources in the lab and will allow us to provide the clinicals with valuable information to help them in their decision-making with their patients. So now we have a couple of minutes for questions. So as we are many today, what I would suggest is to write down your questions in the chat, and I will go through them. So write all the questions you have in the chat, and those that I cannot answer here, we will contact you by mail and answer your specific questions. Okay. So if you have any questions, please write them down. We have a few minutes, so if you might ask anything from what we have seen. Okay, Sandra is asking if I could show the plot with the normal plasmacytoid dendritic cells. So I'm going to go to the normal sample and to show our plasmacytoid dendritic cells. And as you can see, yes, part of them could be expressing either CD16 and CD56. So these markers usually can be presented as activation markers from some populations. As you can see here, you can see this same kind of expression in other populations, like our Tcells. They have a similar expression pattern. Joachim Nilsson is asking if we have any thoughts regarding the enormous change in the granulocyte numbers in the hospitalized patient. And well, as I said, this is a particular patient, so maybe in this patient is going through this reduction, as it is either fighting against the virus and the granulocytes are going down in this way, or either they are being translated or located into different organs, and they are being mobilized from the peripheral blood. But it's very interesting to follow these changes and see how after the infection this population will recover their normal numbers. Okay, I'm going to leave the presentation for a while. So if you have any other questions, you can write them down in the chat, and we will contact you later. And just to finish, again, we wanted to thank you all for coming. We hope that you have found interesting what we wanted to show you today. And to finish the webinar, we wanted to tell you one more thing. Please stay tuned, as we are working in expanding our solution for immune monitoring, and not only for COVID patients. And we hope that we can present you some new and exciting things very soon. So thank you all, and thank you very much.